I would like to welcome everybody of you to this XtraTracks session and would especially welcome Professor Dominik Heider. Dominik, I just hand over to you and looking forward to your excellent talk.
Thanks, Christian, and thank you very much for the invitation. I will share my screen. One sec.
Yeah, it works.
Now you should see my screen.
Yeah,
All
perfect.
right. So today's topic is AI and bioinformatics, fighting the global threat of antimicrobial resistance. And my name is Dominik Heider. I'm the director of the Institute of Medical Informatics at the University of Münster. So first, I will give a short introduction into the topic of AI and biomedicine, and then I will give you some examples from our research, some problems, some solutions that we provide, and at the end, I will give a summary. So as you all know, and probably also heard last week already with the talk on AI and biomedicine, AI is entering all areas of life, including the medical era. And these models, they can, in principle, improve medical treatment, diagnosis, can be used to identify novel subtypes for complex diseases, for example. And they can also give you insights into the underlying mechanisms which will foster intervention studies, for example. And these models can consider all facets of data types, so they can be used for omics data, and I will focus in this talk on this topic, but of course also clinical health records and images. So in our research, and in particular also in the Medical Data Integration Center, we try to integrate all facets of data, including, as I said, omics data, imaging data, but also data from smartphones, for example, and use AI technology, including, of course, like all facets of machine learning, but also approaches such as federated and swarm learning. I will give you some more details later on in the talk, which can be used to use decentralized data. So that means actually that you don't share the data with others, but they stay in the safe harbors, so the hospital, for example, and you can work together multi-center studies without sharing the data. And we are particularly interested in not only applying these approaches, but also to develop new methodologies. So, for example, in the field of biomarker discovery and causal discovery, but also feature engineering, calibration of machine learning models, and what else? And concerning the applications, we use these approaches for infectious diseases, and I will focus in this talk, as already said in the title, I will focus on infectious diseases, but also with other diseases such as cancer, diabetes, and others. And what we also do, we also of course, investigate how these approaches can be really used in translation, and their regulatory aspects come into play because you are entering the field of software as a medical device, and the Institute of Medical Informatics is ISO certified, so we are also actually allowed to produce software as a medical device. Okay, so in my talks, I typically try to summarize the problems with AI and biomedical data with like 5 major problems. And these are in particular the special data types. So in particular, when you work with omics data, you work with sequences, and these are of course not numbers, but sequences. So like DNA or protein sequences. You typically have a small sample size and a high class imbalance, in particular when, for example, you are thinking about rare diseases, but you will also see that this is a problem also in AMR and many other fields. Additionally, as these models, these AI models, should be used in future for treating patients, they need to be interpretable. Otherwise, you don't get acceptance by the medical community and also, of course, by the patients. And additionally, as we are dealing with medical data, which is very sensitive by nature, we have to fulfill data protection regulations. And I mentioned already in the beginning the federated learning approaches, and this is one way to do that. And I will dive into that later on. Okay, so if we consider biomedical data in general, we will see that it's very heterogeneous. So we have very different types of variables, such as, of course, reevaluate parameters such as blood parameters, size, temperature, and so on. But we also have non-standard data types, such as strings or sequences from high-throughput experiments. And these could be, for instance, DNA sequences or protein sequence, but there are also others. And if you ever try to do machine learning on sequences, you will figure out that you somehow have to pre-process the data before you can actually use these models, because sequences can differ in length, and you also need to encode the sequences into numerical values, because at the end, all these machine learning models do some mathematics, and for that you need numbers. And this is also true. I mean, many of you are already familiar with ChatGPT and these transformer models. It's the same story here. So we are not talking about because of DNA sequences, but of normal words and sentences. But still, these sentences can differ in length, and you also have an encoding into numerical values in these models. Although they are called language models, they still do the transformation from symbols or characters to numerical values. So this is problem number one. We have non-standardized data sets. And there's a huge research community working in the field of how to encode the sequences into numerical values. And this ranges from very simple approaches such as sparse encoding or descriptors to fractals, so very deep into mathematics, and many others. And we are also active in the field developing some packages. One of those is Chaos, for example. You can directly download it from CRUN if you're working with R, and we currently have more than 20,000 downloads here. But there are many others, other way to do this. So just to give you an example, sparse encoding, the most simple form that you can do is to basically encode the sequence into binary matrix B, with M being the number of symbols in your alphabet. If it's DNA, it's of course just the four symbols, but you can also do that in principle with text, and then you of course have a larger alphabet. And n is the length of the sequence. So as an example, we have here a very short DNA sequence, ACGTC, and this is now encoded with this sparse encoding into this binary matrix here. You see, for example, at the first position here, we have an A, so the 1 is put here, the rest is 0. Second position is a C, so we have here at the second position a 1. And also at the last one, you see here's another C, and so on, and you fill it. And you also, of course, you extend the alphabet to gap, because in DNA sequences, if you do alignments and these kind of things, you also have this gap symbol here, which could also be encoded here in this binary matrix. And then afterwards, you do a linearization of this matrix. So you basically go row-wise. Sometimes you can also go column-wise, but typically you go row-wise here. And then you put together everything into a long vector. Here we have a length of 20. And this is used then as an input for a machine learning approach. The problem here with sparse encoding is, you see that already, that although we have a very short sequence here, actually the input sequence is very long in comparison. So this is a problem here with the sparse encoding. A second problem is that there is no similarity measure. So you have a 1 or a 0, and there is no similarity between the symbols. But this is not the truth, not the biological truth. So if we go a little bit into deeper and take proteins as an example here, there's a better way to encode protein sequences and also DNA sequences, actually. So another approach which is used a lot are the so-called descriptors. So they are defined as a function from your alphabet, for example, the symbols of proteins, the amino acids, or the nucleotides in DNA, to real values, so that you have the numerical encoding. And typically, physical, chemical, or statistical characteristics of the symbols are used. So for example, the chemical characteristics of the particular amino acid, which is represented by a symbol in a protein sequence. And here's another example. Here you see a short protein sequence, and For example, one descriptor is hydrophobicity. So you basically look up what is the hydrophobicity value of serine and so on, glutamine and what else you have here, and you replace the actual symbol with the corresponding value. So here, another example. These are the hydrophobicity values of these different symbols here, of these different amino acids, and you basically just replace it with the value. And what you can see here is that now you have some kind of similarity. So for example, you see the Q here and the N, they have the same hydrophobicity value. So the similarity between the different symbols is encoded into the data. And this gives you another layer of information that can be used by the machine learning approaches. Another example, another advantage here that you directly see is that the length of the sequence is exactly the same as before. So we don't have this extension here as with sparse encoding. But there are many more. So some years ago, we wrote a review on these encodings here. So there's like hundreds of different ways to encode sequence data into numerical values and also to adjust the length of the sequences to basically make them all the same length in the data set. Besides the sequence-based approaches that are presented to you right now, there are also structure-based encodings. So whenever you talk about a protein, it's not only a sequence, right? So it also has a three-dimensional structure in the cell, and you can also use the information of the structure to do the encoding. So here's, for example, it is the electrostatic hull of this small protein here or small peptide here, and this can be discretized and then also again linearized as a vector and put and used as an input for machine learning approach. And there are many more ways to do that. Again, if you're interested in that, you can also look it up in the review and you have all of the details. So this is ongoing research. Another approach that we are using in our institute a lot is Chaos-Game representation. It's a completely different approach which has some really nice advantages and characteristics. The idea behind that is, and here it's shown for DNA, although you can use it for any alphabet, is that you basically start, in this example, you start with a sequence. In this example, it's 8TGC. So you have this thing here and put the symbols at the corners, and in DNA it's 4 corners. And if you have the sequence ATGC, you start in the middle here and you go half the way to A, to the corner of A, and put a dot here. The second symbol is T, so you go half the way to T, stop here again, you go half the way to G because it's the third symbol in the sequence that we had. And then half the way to C. And so if the sequence is longer, you go further and further and further. And with that, you create such fractals here. So what you see here is the Chaos-Game representation of the human genome, of the mitochondrial genome, but you can do it for any sequence. You could also do it for the genome, but it's then like a different fractal here. And you can see it here for the Iceman, Neanderthal, and chimpanzee. And what you can do with that, you can actually also do phylogenetics. So you could, for example, say, okay, if I subtract the Iceman from the human, I get this result here. And you see that there are some differences, but yeah, not a lot in comparison to for example, human versus Neanderthal. So you see there's more differences. And if you go further to chimpanzee, you see that there's a lot of difference. And the interesting thing about this is that what you can actually see here is that the Chaos-Game representation creates fractals. And these fractals are very specific to the organism. So you can do that, for example, also with the genome sequences of bacteria. And just by looking on the fractals, you can identify E. coli or what else. So depending on the phylogeny, so the closer they are in phylogeny, the more likely they look similar to each other in these fractals. And this is very interesting and can be used as well for machine learning approaches. As said, you can also do that with any other alphabets. for example, protein sequence where you have 20 symbols. Then instead of 4 corners, you have 20 corners. So you can, I mean, not really see it because it's too small, but with these protein sequence and these 20 corners, you build these snowflake structures here, which can also be used for machine learning or phylogeny as well. And you can do that. You can also do that with text, with books or whatever. They look then a bit different, but it's the same story. And it's a very nice approach to encode the data, because really one of the biggest advantages is here that it's completely independent from the length of the original sequence, because the dimensionality of these figures are always the same, no matter how many symbols a particular sequence has. Okay, so I already started to talk about omics technologies, and I said that you get up with DNA or protein sequences, but you can do actually a lot of different things. You can, for instance, look at genomic sequence, then it's called genomics. You can look at gene expression data, it's called transcriptomics. You can also sequence populations, for example, the gut microbiome and things like that. Then you can either do amplicon sequencing, that means that you sequence a particular part of the genome, and that for typically it's the ribosomal RNA that you use there and to do the taxonomic annotation. But you can also do metagenomics and metatranscriptomics. And actually nowadays you can use basically any word and put an omics behind that, and it's very likely that it exists. So there's a lot of other omics technologies right now. There are also many different platforms that have been used in the past and that are currently used, and these numbers are a bit outdated here because they are taken from a review from 2017, but just to give you an overview of the different technologies that are in place. And we have experience with almost all of them, but some of them, for example, the 454 technology from Roche is not used anymore, but we used them in the past. Nowadays, it's basically Illumina, Oxford Nanopore, or PacBio. These are the main players here, but there are also some others that have been used in the past, like the QIAGEN Gene Reader and so on. And in my institute, we develop different tools and methodology, as I mentioned in the beginning, to work with these different omics technologies, such as for multi-omics integration, embeddings, differential abundance analysis, microbiome analysis, or comparative genomics. And we also investigate in AI development, in particular for heterogeneous medical data. Okay, but now coming to the actual topic, which is AMR and how AI could help to fight AMR in the future or nowadays as well. So as you might know, AMR is a really huge problem. So there have been estimations by the World Health Organization that in 2050, there will be around 10 million deaths yearly because of antimicrobial resistance. And in comparison, for cancer, it's estimated to be 8.2 million. So it's becoming a larger and larger problem. And already today, we have around 1.3 million deaths due to AMR. And the main driver of this is, of course, the mis and overuse of antibiotics, not only in humans, but also, of course, in animals and what else. So what we did in the past, together with colleagues from the University of Duisburg-Essen, was the largest sampling campaign in Europe for freshwater lakes. And this is very interesting because there has been a lot of research in wastewater, and I will also give you an example on the next slide, but this was the largest one that was carried out in freshwater. So they actually went to, and you can see all the different sampling sites all over Europe, where they did a sampling with a standardized protocol. And then in the next step, they did also the metagenomics sequencing, and then we analyzed the AMR, the resistome of these freshwater lakes to get an impression of how far resistant genes are spread in Europe in freshwater. And this is what I said. This is a research done not from us, from others in wastewater some years ago. And what you can see here is the different sampling sites on the y-axis from wastewater samples and different antibiotics ordered by the time they have been invented or used in practice. And what you can see here is that uh, of course, over time, there is more and more resistance shown here in red in these different wastewater samples. So with more use of antibiotics, there's more resistance, of course. And here, for example, I just highlight one tetracycline, for example, which is a broad spectrum antibiotic, which is used a lot in practice. And you can see that there's a lot of resistance in wastewater, which is actually clear, right? Because if you take antibiotics and then you go to the toilet, it's washed out and goes into the wastewater. So what we tried to figure out, we wanted to do the same, but with fresh water to see how much AMR is going into the environment. And here you can see a similar plot. You see here, these are not all sampling sites, but some of the lakes here, see the lake IDs. And here you see the different antibiotic resistant genes. And you can already see that there's a lot of resistance in these different lakes. You can also see the different countries here on the top. But you see, of course, that for some it's already very tricky, and for some others there's no antibiotics at all or no AMR at all. And here I highlighted 4 because I use them on the next slide again. And there's also the tetracycline here. So you see that for some lakes, here in particular this, for example, you see already a lot of AMR in these freshwater lakes. But we also highlighted some others that are used a lot in practice. And like cephalosporins, for example, and also others that are not frequently used, but are rather considered to be a last resort antibiotics. And here are the 4 again, now put on the map of Europe. And you can see that, for example, the cephalosporin here, there's a lot of resistance already found in these different sites. For others, it's much better. Like, for example, here you see that there's a little in Germany and here, but it's not comparable to what we found with this antibiotic here. We also tried to find association with livestock farms, and we found one, but they were like slightly not significant, so we could not claim this that there is a connection, although it already looks like, but it was simply not significant in the statistical analysis. Okay, so what we did next was, okay, if we know that this is an ongoing problem, and in particular, if you have nosocomial infections in the clinics, it's a really big problem if antibiotics are not working anymore. So the typical way to do antimicrobial testing is in the lab. And this is time consuming, of course. So we ask ourselves, okay, is there a way to make this easier, more precise, and of course, faster? And in our BMBF project, we tried to develop a prediction pipeline that was based on whole genome sequencing. So it was the genomics part, basically, of different isolates from patients. And we tried to develop machine learning models that use the genomic sequence as input to do a prediction for antimicrobial resistance. And as you can see here, we collected, or the colleagues in Gießen collected in microbiology lab, they collected like roughly 1,000 samples from patients, and we also collected 1,500 samples from public data. And we also we took the phenotypic information for 4 antibiotics to develop these models, and we tried out different approaches to see how it's working. So we basically did a cross-validation here with our own data and then evaluated it with the public data. And what you can see here is that you see precision and recall here. You see that it's actually working quite well, in particular for some drugs like CIP, for example. It's very high values here. For others, it's a little less, but still you see that it's working. for like almost all classifiers. It's like not like the same, but they're all quite good as you can see here. And what we also did, so we looked into the underlying SNPs that we found to be important for the prediction. So we also tried to make these models explainable, and we found a bunch of things that are associated. Oh, sorry, associated with the different drugs. However, these mutations or these SNPs are not known to be related to resistance. And there's one very important fact, and that is that most of resistant genes are not in the genome, but in plasmids. And if you do the genomics, you don't get this information. So we simply don't have the information on the particular resistant genes, but what we see here is what we called secondary mutations. So we think that they are compensatory to compensate for the fitness loss for the carry up of the plasmid with the antibiotics resistant genes. So that is our interpretation of that, and the colleagues in Gießen are currently trying to figure out what these SNPs do in these particular genes, but it's very hard to find out because we see that they are somehow important for the resistance, but they are not directly related to the resistance mechanism. Okay, so additionally, there is a very interesting fact that happens not only in bacterial resistance, but also in viral resistance or in cancer, and this is called cross-resistance. So that means, I give you an example from HIV because it's easier, because typically when you do antibiotics, the bacterial dies, and but in HIV you cannot really cure someone, you can just treat it and reduce the replication of the virus. So cross-resistance means that for example, you treat a patient with a particular drug, and then after some time, there's resistance coming up in the virus, but this resistance is not only for the drug that has been given before, but also to other drugs that are currently unseen by the patient. And this is called cross-resistance. And due to the fact that we know that this exists, we ask ourselves the question, could we use the information on cross-resistance to improve the models and make better predictions? And this is what is called multi-label learning. So in contrast to the typical situation where you have an instance, so for example, a protein sequence or a genomic sequence, and the label resistant against drug A, for example, you can, in this multi-label approach, you can combine one instance with a bunch of different labels in one model. And with that, you get the interdependencies between the different labels, and they are modeled directly into the model. There are many different approaches of how you can actually do that, which are listed here. And I don't want to go too much into detail here, but on the next slide, I will give you one very easy example that you understand how you could do that in practice. And these are the so-called classifier chains. They are very easy, although we see on the next slide that they are not the best performing one, but an additional model that is based on this approach made the best predictions. But okay, let's start here. So how is it working? So in this example, we have 5 classifiers for 5 different drugs. And the first classifier for drug 1 gets the normal sequence, for example, this genomic sequence or the protein sequence as input, and makes a prediction for drug 1 resistant or susceptible. The second classifier uses again the sequence information plus the information from the first classifier for drug 1 to make a prediction for drug 2. The third one again gets the sequence information plus the predictions of the first 2 classifiers to make the prediction for the third drug, and so on and so forth. So they are all put into a chain. And this is why this is called classifier chain. And the problem here is that the order of the classifiers affects the final prediction very strongly. So what people typically do is they extend this model to a so-called ensemble of classifier chains. So instead of having just one classifier chain with a predefined order, they permute the order and then they built an ensemble of these permuted classified chains to make a prediction based on majority vote. And this actually turned out to be the best approach. So the ECC is the ensemble of classified chains, and you can see here, it's shown here in gray, that they perform better than the other approaches that we can see here in the other colors. And this is also significant here, with exceptions for some of these drugs here. But in the most cases, it's significant. Okay, next thing. So as you have seen, we restricted our analysis to 4 antibiotics, but there are so many. So the question is why? And the problem is that the data sets are limited. So for some of the data sets, we have only little information about resistance, or only some strains that are resistant, or we can see only resistant strains and only have some that are susceptible. But we ask ourselves the question, can we still train a model although we have this limited data set? This is problem number 2. And I want to show you a real-world example, and then we'll dive into how we use that approach in the AMR models. Okay, so my question is, who of you play an instrument? And the typical situation is that when you were a kid, your family or your parents forced you to learn an instrument, let's say the piano. And so when you started to learn that, you learned, of course, to play the piano, but you also learned to read a melody from paper. You also learned other stuff that are related to music, and then you ended up with playing the piano. Now you grow up and you decided to learn the violin. And now again, of course, you have to learn how to play the violin. But the things like reading melody from paper or other related stuff to music, you don't have to learn it again. So it's already there. So you don't start from scratch. And this is the same idea that we followed here. It's called transfer learning. And the idea here is that we train a model here symbolized as a feed-forward neural network, but could be typically you do that with neural networks, but it could be any kind of architecture. You train it on a data set where you have a lot of samples. So for example, for one of these 4 drugs I mentioned before, and then you take the drug where you have less samples and fine-tune the model that you developed here. So instead of starting from scratch to learning a model from scratch, you start with this one here and then change it a little here and there with your new smaller data set, and then it's fine-tuned for the second data set. And this actually works quite well, otherwise I wouldn't show that to you. So here you can see the different drugs again. And here are the results. So in blue are the original results learned from scratch. Now we don't have the precision or recall with the MCC, which is a metric that is typically used when the data set is imbalanced. But the essence here is that we can see a significant improvement here in yellow if we use transfer learning for these data sets here. And this is not only true for our own data set, but also on the public data set. Although, of course, because of batch effects and what else, the MCC is a little lower here compared to our own data set. How do you do that? So here's an example for a CNN that we used here. And what you typically do is you take the CNN and then you freeze some layers, and you can just play around a bit which layers to freeze. And the others are then again trained with the smaller data set. And with that, you get this fine-tuned model that is not overtrained to the small sample size. And we use that approach, this transfer learning approach, also for other things. So you can also you cannot only use that for training data set or training models for small data sets, but you can also use that between technologies, for example, to transfer knowledge from bulk sequencing experience to single cell experience and so on. So there's a lot of variety of what you can do. The only thing that you have to keep in mind is that the original problem needs to be as close or similar to the problem that you try to solve with this. And that's all. So this is how you can use the transfer learning. What we also do, as said before, is biomarker discovery. So, for example, differentially expressed genes in cancer or parameters that are associated with a particular disease. And in medicine, this is typically called biomarker discovery. In computer science terminology, it's called feature selection. So the idea is to identify the relevant features for a particular problem. And the reasons are simplification of the models, of course, to get a better generalization, faster predictions. If it's less data that need to be processed, of course, it's faster. And if you want to interpret this model, this becomes much easier if you have less input dimensionality. The problem here is there are many methods out there which can be used for feature selection or biomarker discovery, but they all have specific biases. So what we wanted to do is to improve stability of the feature selection approach and reproducibility. For example, many people in practice use random forest for feature selection. So they use the importance analysis of the random forest, but this is biased, for example, to features that have more categories. And as medical data is typically heterogeneous, so you have biomarkers, you have sex, age, what else? The random forest importance analysis typically prefers features with more categories. So in this case, for example, a blood parameter that is a real value compared to sex, which is typically binary in these data sets. So what we did, we basically tried to combine different feature selection approaches again in an ensemble. And we selected the approaches in a way that the different biases are compensated so that they are basically in the opposite direction and with that are more or less robust. And we provide a web server here, which can be used where you can just upload your CSV file and then everything is done automatically and it identifies and ranks the different parameters that you can use then in subsequent machine learning analysis. But we also provide an op package which has a little bit more functionality. But this is just if you want to do it for example, in a pipeline or something. And it has been used a lot. Here is an example from a collaboration with the Lung Institute in Marburg, where they used the EFS approach to identify genes that are associated with malaria. And here on the left, you can see, for example, the ranking of EFS for the different features that have been used here. And you can see here the different methods that have been used that are aggregated. And what you see here, what you see here, for example, is that if you have used, for example, the random forest here, which is here, you see a different feature selection than, for example, for this method here. So it differs. So and this is what it makes it a bit tricky if you want to interpret it at the end. So what is going on there? In the biology, having a robust approach is much better than having like not a random selection, but a bias selection. But we can also go deeper. So we also, in the recent years, we invested a lot in research in causal structure modeling. So the underlying idea here is that you not only identify the important features in terms of predictability so that they can be used in machine learning for diagnostics so that they are good predictors, but also to find causal relationships between the different features. And here you can see two examples from two recent publications or one archive paper from us, where we can see with these arrows the causal direction of the relationships between the these different parameters. And here, for example, in this model here, the phenotype was depression, and here we can see that there are some, for example, BMI is a causal factor for depression. This is already known, but there are also here, for example, from the microbiome that we identified with this approach. Yes, data imbalance, I mentioned that already in the context of small sample size. Here again, you see the entire data set with the different antibiotics here from our study. And you see that for some, I mean, for these, the proportion is 50-50, but for others, it's much worse, right? So here there's only a little some isolates are resistant. Here you see that most of the isolates are resistant. So it's not always 50-50. And this is typically a problem in machine learning. So there are many approaches of how to handle that. If you have a data set which is very huge, like millions of samples, you could just remove some samples from the majority class to balance it. But typically in the medical scenario, we have small sample size. So removing samples is not the best way to do that. So this is problem number 3. And there are many statistical approaches that can be used that in the field of data augmentation. The most common approaches are SMODE, Addison, and upsampling. But there are also others like generative adversarial networks, so basically neural networks that can be used for that. And we did a small study where we analyzed small imbalanced medical data sets to analyze which of these approaches that are commonly used performs best. And just for the sake of completeness, I want to briefly explain how these approaches work. So SMODE, for example, is a synthetic minority oversampling technique. So the idea here is that you take the minority samples here and you select one of those. You select 4 neighbors. If K is 4, of course, you can also adjust this parameter here. And then you select one of these neighbors randomly, and you put an edge between them and put in the middle of the edge or somewhere left or right. This is also a parameter that you can adjust. You put a synthetic data point. So you're basically in between 2 existing data points. So this is the whole story for SMODE. Addison is similar to SMODE, but gives higher weights to difficult data points so that they're more often selected. Upsampling is just basically copying an existing data points, and Gaussian noise upsampling is upsampling plus adding noise. And as said, the GANs are a neural network architecture for data augmentation. Okay, so how is Gaussian noise upsampling working? So here again, we see samples from the minority class, and we have a parameter that can be adjusted. So that is basically a radius around these data points to create the new ones, and they are randomly created there. And with that, you have the upsampling. And you can see an example just from one data set. In the study, of course, we used more, but the results are more or less the same. So I just used that one here. You see that it's imbalanced. We have 225 patients who survived breast cancer and 81 who died. And the upsampling approach, SMODE, Addison, and the Gaussian noise upsampling is visualized here just 2-dimensionally with the PCA, just to make it on the slide. Of course, you can also, in the classification, we used it in the original dimensionality. And what you can see here actually is a problem that sometimes occurs with SMODE because of the idea that you combine 2 existing samples with a line and put a sample on this line. You could sometimes, if the data set is small, you could enter up with such like a kind of grid in the data set, which has some effects and bias as well if you train on that. So with this small data set, it turns out that SMODE and Addison is basically the same. It is sometimes a bit tricky, and it doesn't work well in all data sets. However, Gaussian noise upsampling often outperforms Smote and Edison, although it's a very simple idea, but should be used first. But typically you can also, of course, try out all the different other approaches. Okay. Okay, let's imagine we now have such a prediction model and we want to use that in translation. There are some other problems, but one problem that comes first is that you need acceptance by the medical community. Otherwise, they won't use that. And if you want acceptance, these models not only need to have high accuracy, but they also need to be some kind of interpretable. There are many different ways of how to make machine learning models interpretable, and I want to give you just one example that is a first step and is important to get this acceptance. So whenever people use machine learning, they assume that the output, or like many people do that, not all, of course, but many people assume that the output of a machine learning model is a probability, but that is not true. That is not true. It is actually a score. Of course, if you use a Bayesian classifier, that is a probability, and this is also true for logistic regression. But if you take a random neural network, for example, it's not. It's a score. And to actually get a probability, there are methods out there called calibration models. And these calibration models take the uncalibrated machine learning scores as an input and gives you probabilities, which are then easy to interpret by the physicians, for example. So that means that if you have in a model, for example, that gives you a 0.6 for cancer, it doesn't mean, or it does not have to mean, that this is 60% probability. After calibration, it should be 60% probability, but before it could also be 70 or 50 or what else. Okay, this is problem number 4, interpretability. And here you see a plot, you see the machine learning scores, and here it's clear that it's not a probability because it ranges from minus something to 6, right? You can see the distribution of the scores for the controls and the cases. And as I said, there are many different ways to calibrate these models, and I will to give you some examples and an approach that we developed some years ago. So the most straightforward way to get that is called equal with binning. The idea is that you put a grid or bins over these distributions here, and then whenever you want to have the probability for a new sample, you basically look, okay, the new sample is, for example, here in this bin, and then I have the fraction of controls and cases, and with that I can estimate the probability. The problem here is that in these areas here where you don't have too many samples, all these estimation won't get significant. So you cannot really say that the probability is very robust. Some years ago, some researchers published a barbecue. It's the same idea, but instead of equal width, you go equal frequency binning. So that means that when you have more samples in an area, you make the bins smaller. And if you have less samples, you make them bigger. The problem here is, again, these areas here where you have very low frequency, it's not working, it's not significant. So we thought, okay, is there a way to make it better? And our idea is basically driving this bin idea to infinity. So instead of using bins, we actually calculate the density function here. And from the density function, we can actually calculate the p-value. So the same as if you would do a t-test manually. And what we do here is we first estimate the distribution of the scores. So for example, this looks like roughly normal or a little bit, I don't know, t maybe. So we estimate the distribution, and then we calculate the actual p-value from the estimated distribution. And here you can see the results. So we did that on simulated data as well as real world data sets, acute inflammation, breast cancer, cancer survival, and some others. And you always see for each data set, you see two plots. The first one is the calibration error, ECE. It's a metric with which you can estimate the calibration error of your model. And what you can see here is that all models here on the x-axis, you see histogram binning, barbecue, and gas. So this is our approach. You see that they do what they should do. So they decrease the calibration error. The second plot here is the AUC. So this is a metric which is used a lot to show how good a model can do the actual prediction. And what you can also see here is that when you calibrate the models with these approaches here, the AUC drops. So the model is not as good as before in terms of classification accuracy, so to say, which is not true for gas when we estimate the p-values here. And you can see the similar results also for the real world data set. So for example, here you see again the calibration error drops, but also the AUC for the binning approaches, which is, I mean, there's also a little drop here, but which is not true for gas here. This here you see other examples. It's the same story. So what we can see here is that if you use our approach, you get a reduction in calibration error, but the prediction performance of the resulting model stays the same. And this is what we want to have, right? So we don't want to have the trade-off between interpretability and performance. And this is also available as an R package, and you can just put it in your pipeline after your machine learning model, and then the model is calibrated. Okay, coming to the fifth problem already. So everyone is talking about big data also in medicine, but if we are honest, medicine is not in the era of big data yet. So typically, data sets are small and people have to do multi-center studies to get enough data sets. So they work together, also internationally, to get not only for rare disease, but basically for everything, to get big data sets that are representative and can be used as an input for machine learning. So what people did in the past and used in the past was to store the data in a cloud, right? So to anonymize, for example, data and to put it in a cloud. But for some data, this is not possible. So genomics data, for example, you cannot just make it anonym, right? So this is very tricky with respect to data protection regulations. And another thing that is also, well, not because of regulation, but which is also very important. So if you upload your data to a cloud to be shared with others, the data is shared. So you lose data ownership, right? So you have to trust them that they do only what they said they will do with that, but you cannot guarantee that. And a solution for this problem is federated and swarm learning. So this is problem number 5, data protection of sensitive data. And the idea of federated and swarm learning is essentially the same. The underlying network structure differs in these two approaches. So here on the left side, this is a figure taken from a paper that we published 2 years ago in Lancet Digital Health. You see the federated approach, and here you see the swarm learning approach. And the difference is here that here in this example, we have 4 hospitals who want to work together. And we have a central server here in the middle in the federated approach where the models that have been trained on the local data are aggregated into a final machine learning model. So the idea here is we have local data. We don't share the data, but we train a local model, for example, a neural network or random forest or whatever. And then we share, instead of the data, we share the model with the others, and all the others do the same. And then the models are aggregated. And depending on what machine learning model you have, this aggregation step differs from approach to approach. And then after aggregation, the final model is then broadcasted back to all the clients. So they have all the final model that have been trained on all data, but the data never left the safe harbor. And the swarm learning approach is essentially the same, but instead of having a central server for aggregation, every client acts as an aggregation server. So we basically have a peer-to-peer network. The advantage here is that if you, for example, consider an international collaboration here between 4 parties, let it be, for example, in Germany, the US, Australia, and China, the central server needs to be somewhere. It could be Switzerland, for example, right? But you need some kind of a trusted party here that does the aggregation. Here, everyone is equal. So everyone shares the model and aggregates the model. So there's no central server necessary. And this is the advantage of swarm learning, although network communication is here a little bit more because you have these broadcasting to all partners. Okay, so how does it work? And I want to give you here a very easy example, the linear regression. Although the linear regression is not privacy preserving because of outliers, for example, it's very easy to explain federated and swarm learning on this example. This is why I show it to you. So if you use linear regression in a federated approach, just keep in mind that it's not privacy preserving per se. Okay, here you see the example with the 4 hospitals again, and the only 2 parameters you need to describe a linear regression model is the slope and the intercept. So here you train a linear regression model on the local data, here again, and again, and you have these different parameters. So the 2 parameters at each side are transferred to the aggregating server and then averaged here. So we have the average slope and the average intercept, and this is the final model. And you can also mathematically show that this is asymptotically the same as if you would aggregate the data and train the linear regression on the entire data set. Here's another example with the random forest. This is even easier actually compared because you don't have to do any averaging or anything like that. So a random forest is basically a set of decision trees that are combined via majority vote. And here you train these decision trees, so like 500 or what else, on your local data, here again, and then you transfer the random forest, all the decision trees to the central server where they are just put together, and it's majority vote, so you don't have to do anything, just do the majority vote on all the trees from all the sides. That's it. For neural networks, it's a little bit more complicated. Here you see in this toy example, you see very small networks, so they all have to have the same topology. And you train these weights here on your local data set, and then you send the weight vector to the aggregating server where an averaging is done over all these different weights, and they are sent back, then trained again in the next epoch, sent to the server, aggregated again via averaging, sent back, forth, set off, and until it converges, and then you have the final model. Okay, so with that said, federated learning and swarm learning can be used in these collaborative settings without sharing sensitive data, which is really nice. Instead of sharing the data, model parameters are shared and aggregated. And this can be used for like basically any kind of machine learning model. However, you have to keep in mind that it's not per se privacy preserving for all these machine learning models. So for example, linear regression, I already said that you might detect outliers, which are patient basically, or support vector machines. Of course, the support vectors are patient, so it's also not privacy preserving, but also KNN, of course, and there are many other examples. So whenever you want to make sure that it's privacy preserving, you have to add another layer there, which could be, for example, secure multi-party computation, homomorphic encryption, or what else. So, but by combining these different approaches, and in particular, also using federated learning, where the data can stay in the safe harbor. You can guarantee that there is no data leakage and everything, that the privacy is preserved. I'll also give you an example of a particular tool for a particular application that we developed in the Institute for differential abundance analysis. This can be used, for example, for gene expression or microbiomes, because nowadays people consider to be the microbiome of the gut, for example, of a patient, to be a fingerprint. So that means that in theory, you could identify a person by its microbiome. So, and with that said, it should make sense to use a federated approach for differential abundance analysis as well, if you want to do it in multi-center studies. And we developed a software, a framework for that, which is called Glinder, which is based on Linda. Linda is a software that, or a method that have been developed by others, but we made it federated. And with that, it can be used in a decentralized, privacy-preserving way. And here in these plots, you see the analysis of the data sets. And so on the y-axis, you see the p-values for the identification of the different taxa with Linder. So this is our ground truth. And on the x-axis, you see the different approaches, so the p-values of the different approaches. And here in the first plot, you see Glinder, and you see that they are basically all the dots on the diagonal line. So it's very close to the predictions of Linder, so it works over all sides without sharing the data. And this is the ground truth was set on pooling the data and then do the Linder analysis. If you do meta-analysis, you see that it's not working. So you would not identify any significant taxa here. And also the individual peers here with Linder, you see that it's not working, right? So the data sets at each side are too small, basically, so you don't get significant hits. So it's not working. And only by pooling, which you should not do because of the privacy issues, but also with Glinder, which is actually not federated, but a swarm learning infrastructure that is used here, you can identify the taxa and everything is working without sharing the data. We also developed with colleagues from a big EU consortium. We developed ASPLink, which is the federated version of Plink, which is used in GWAS analysis a lot. And here we could also show, as you can see on the next slide, that in particular, if in the imbalance scenario, you see that in blue, the p-values of Plink, the ground truth, and ASPLink, our federated version, are in line here, while the red dots symbolize the meta-analysis p-value, and you see that it's not working. So we would not detect significant SNPs here with meta-analysis. Okay, to sum it up and to come to an end, if we want to use AI in biomedical data, we have 5 problems that need to be addressed with new research methodology. We have special data types, in particular DNA and protein sequences. We have typically small sample size and high-class imbalance. The models need to be interpretable to get acceptance by the medical community and, of course, also the patients. And data protection regulations are super important in medicine, of course, and need to be addressed with these approaches. And yeah, coming back to a slide that I have shown at the beginning. So what we do is to try to combine the data, use all and develop all the methodology that is needed to address these 5 big problems. And we apply them not only in infectious diseases, but also in any other kind of diseases and with our experimental partners. And with that, I want to thank you for your attention. Of course, all the people working at the Institute of Medical Informatics, because I mean, they are actually doing the work, the daily work here. And yeah, I'm now open for questions. Thank you very much.
Yeah, Andreas speaking. Dominik, thank you very much. That was a really great lecture. And sorry, my background, I'm on a lab fair in Basel, which they are just dismantling. So that's why there's a little bit noise in the background. Sorry for that. And also, by the way, here AI was a big topic as well. So it's everywhere really. Also in the pharma industry, in different topics, and even in the lab, right? I have one question. I mean, that's the usual question I always ask. I mean, you presented these 5 problems and quite a few solutions to each of those problems. How did already, you know, did some models already do the way to the, let's say, standard usage? Could you show that they work and that they are accepted by a specific community? Is that's thank you. That's of course always very interesting, right? Because there's a lot of research in your topic and set in a lot of other topics as well. But finally, it's always like, does it work? Can we use it, right? Yeah.
Yeah, that's true. I think the problem with the actual translation and bringing it to the bedside is the regulatory aspect, right? So whenever you want to use software or AI models really at the bedside, you have to fulfill all the regulatory requirements as software as a medical device. And this is a way research institutes typically don't go because it's too expensive, right? I mean, we are certified, so we could do that in principle. We're allowed to do that. We have an ISO certificate. However, in practice, there are not so many incentives to do that, to be honest, right? Typically, I would say the best way to do that is to collaborate with industry, because first they have the money and they have the time to do that, right? I mean, they also have to do a lot of work, but they have the processes to really go and to prepare all the documentation for FDA and the European counterpart. And I think this is nothing a research institute can really do, because we have a lot of fluctuation in the people working in the institute, because people do their PhD, then they leave and so on, right? So this is very tricky. And as said, in theory, we could do that, but in practice, it's very unlikely.
And did you find already partners for certain approaches or models? Or is it more like that you give, like in this talk as well, the ideas, what people have to think about, and then they say, thank you very much, you're publicly funded. We are happy that the state is doing that, and we are using some of your ideas, which you publish, of course, and use them, and then maybe make a product out of it, right?
Yes, actually, it's typically that way, because we also try to make everything open source so that the people can reuse, and that someone at the end will bring it to the bedside. But there are also examples in particular when there's no direct involvement of patients. There we have some collaborations with industry where we developed software for like machine learning models for example, like peptides and what else, that are used in industry already. So, but there you don't have, they are not used as a medical product, but they are used, for example, in their processes, in for example, like synthesis of peptides and these kind of things, where we did some optimization of processes and so on. So this is where it's actually used in practice. So it's not all theory. But for translation in really for patients, it's very, very tricky and a very long way to go, which is typically not done by the research institutes themselves.